Okay, so this is my model, and first for the required components, the model is sturdy enough to be handled without breaking. Then the model is 40 centimeters long, and the DNA molecule does depict all 4 types of nucleotide. So here we can see that here is A, T, C, and G, and we can see them here as well. Then the DNA bases are arranged in complementary pairs, so A is always with T and G is always with C, and here we can see that as well. And then the sugar phosphate backbone of the 2 helixes is on the outside and the bases are on the inside. So here we can see that the sugar phosphate backbone is on the outside and the bases are on the inside, and in this model as well, this is on the outside and the bases are on the inside. Next, the bases radiate from the sugars, so here we can see they're connected to the sugars, and here we can see that as well, they're connected to the sugar phosphate backbone. Then there are 10 nucleotide pairs, so here there are 10 and here there are 10. Next, the diameter of the double helix backbone is consistent, so here we can see how this is consistently going, and here as well it is a consistent light line, and here we can see the twist. Then there is a key to the parts of the DNA shown in the bottle, so here is the key for this one, where we can see, and these are the same colors for the nucleotides, and here we can also see how everything is labeled. So here's the base pair, the nucleotide, the nucleic acid, and everything is labeled. Then for the optional features, the blueprint does match the completed product. So here we can see how they're like the same. And then there are 2 nucleotide pairs per complete helix turn. So here we can see there are like the 10 pairs and the turn. So here we can see like where it turns and where it turns. And then there is the correct number of hydrogen bonds between bases. So here in the key, the red line means the double bond and the black line means triple bond. So in A and T there is a double bond, and in C and G there is a triple bond, and here it is represented with a line. So here we can see 3 lines and here we can see 2 lines. Then the complementary strands are anti-parallel. So here we can see at the end there is the sugar end is 3 and the phosphate end is 5. And next, the twist of helix shows 2 strands in parallel rotating around the central axis. So here we can see the twist shows that it is rotating around the central axis, which is like the base pairs. And next, the model has artistic merit. So that is why I like colored everything, and I made this with like paint. And then the description of the protein and diseases associated the actual DNA sequence modeled. So here is the disease that I chose, which is breast cancer. So here is the protein of the breast cancer and a definition of what it is. And here is the disease that is caused from that, which is breast cancer, and it is a small description. And then this is the protein that is drawn, and here is the disease that is drawn. Thank you.